Hello, my name is Rob Durham,
And I'm John Chappell.
and you're listening to another edition of the Gyrolab Spin.
Today's podcast is a special edition to celebrate the 20 years of the Gyrolab technology. 20 years ago, we released the first CD or compact disc and the first instrument. So back in 2003. So we are celebrating 20 years.
Yeah, time flies when you're having fun, doesn't it, John?
It does indeed, and I mean I've been using this technology for most of that period actually.
Yeah,
So, and I, as a customer, always found it a cool technology, and really loved the development speed, and really implemented it back in first in 2006 is the first time I
yeah,
implemented the
and
system.
I think you and I were working together on a project that I was doing at Pfizer, and you introduced me to the technology, and I think it was around 2007, 2008, and really, thought it was a cool technology,
Yeah.
and so it's great to see that it has grown over the years and become a very well-established method in the bioanalytical space.
Yeah. So today's edition, we really want to highlight the global user meetings we've had, because as a customer, I really liked the fact that Gyros gave us the opportunity to come together and discuss our applications, but also improvements in the instrument, and things that the customers wanted. So the user meetings for me have always been a key part of the Gyrolab use, and really why users like to use the technology.
Great. And so to celebrate our 20th anniversary, what John and I'd like to do is take you on a little trip around the world where we hear about all the wonderful work that our customers are doing on the system.
So this year we've had 4 global user meetings. So it started off in Europe, where we had a meeting in Lisbon. Followed in the last month, we had meeting in Tokyo in Japan. This meeting is a regular meeting, but this is actually the first meeting that we've had since the COVID-19 pandemic. So it was great to have the opportunity to meet the Japanese customers, and there is a lot of loyalty and excitement in Japan following the meeting, and we discussed many applications, and the Japanese users are really great users of the platform. And then we moved on to Shanghai in China, where we actually had the first user meeting in China. What we're finding is a lot of European and US customers are starting to move projects to China. So as well as the established kind of pharma industry in China, we're finding that now that the CROs are starting to get work from our customers, and so we're starting to have to support and help offer support to China, and it's really exciting to see the growth in this region.
It is, and I know that we've returned to going to conferences again as well, and at the conferences I've been to, you see a lot of growth in the Chinese CRO business. So
Yeah.
it's a great space.
Yeah, and it was a great meeting, and it was the first one, so we expect it to have this meeting every year, just like in Tokyo as well. So we'll continue now for that to be a regular event.
Excellent.
Yeah.
And then you brought it on home to Philadelphia. At least it's home for me, not for not for everybody out there, but brought it home to Philadelphia, where we again, I think, had the second real meeting since the COVID break, and I think it was just a really well attended event.
Yeah, it was great to meet the US customers, and I think again it was a great event, and we got great feedback, and
Yeah.
and it is kind of the DNA of Gyros, that we're very customer focused, and these user meetings are where we get suggestions for improvements or for new applications. I mean, I raised the potential for cooling of samples and reagents on the system back in 2009 at a user meeting,
Yeah.
and that was implemented onto the Expand system. And I know you requested Affinity
Yeah,
back in
exactly.
the day, and there was a
Yeah.
very young looking Rob video that's
Yeah, yeah,
circulating out there
that's right.
where you specifically asked for Affinity. So it just shows you that that they listen to customers and really look for things to improve the system.
And I think if anybody out there has not had a chance to get to a user meeting, you know, look for the opportunity to come join your colleagues who are fans of the technology or people who are just exploring the technology to see how that might enable the work that you're doing. And so really, again, it's great to have a global presence and to have seen how the technology's grown throughout the world.
Yeah, no, it's very exciting. So we thought we'd summarize some of the discussions and some of the presentations that were had at all four meetings, and some of the main topics that were discussed amongst our customers. So I mean, one of the big things that we tried to do was discuss maintenance procedures, particularly to try and harmonize things like using hot water wash, particularly to help with the cleaning of the wash station.
Yep.
And this impacts really heavy users, which we're finding now. We have a lot of users using the system very regularly. So it's just a good practice and very quick and easy to do.
Yeah, and I think if you have any questions about that, I mean, the advent of using pH 11 to really deal with carryover issues on the system, led to the creation of another potential problem, and that is plugging up of that wash station. And so we know from lots of experience that if you allow that wash station to become clogged up, it's not going to clean effectively, and then you're going to run into carryover, all kinds of difficulties with your assay. And so if you have any questions about maintenance, you should really reach out to your local field application support scientist, and they will help you mitigate any problems you have with your maintenance.
Yeah, and I think another important part of the meetings was to discuss assay troubleshooting, so the tricks and the things to do to actually develop assays. And although the system is very easy and quick to develop methods, there are things, particularly we discuss things like competitive assays, but also other formats and how to develop alternative to the mainstream assays, and kind of share our experience. Things like column capacity, with bridging assays, you have to reduce you don't saturate the column, you reduce the amount to ensure that you form a bridge. So there are different things with the Gyrolab to consider because of the microfluidics and the fact that we're moving the kind of assay over the column.
Yeah, exactly. And I think their hallmark of our meeting over the last several years has been the development of these workshops that help give customers a chance to come in, learn some of the best practices from the application scientists that are doing this day in and day out, and offers them a chance to ask questions. And I heard a lot of discussions at the U.S. meeting where customers were bringing their real world problems and to have those discussions at the meeting and get a lot out of it. And I think it's one of the things that really adds a lot of value for our customers. And everybody's going through tough budgetary times, and so going to another meeting can seem like it's discretionary money, and it's not always been made available, especially if you're going through difficult economic times.
Yeah, and this is important why we always include basic and advanced training as part of our user meetings to give customers the opportunity to learn new things, but also to kind of share some of the new assay formats and how to interpret some things like viewer profiles, for example. So I think we offer this training as part of the user meeting, and I think this year we completely refreshed the training because we did get some feedback that the training sessions were kind of the same between different years. So we tried to bring new things this year, and so we do listen to customer feedback to ensure that we were giving some fresh approach to the training.
Yeah, I think I mentioned earlier the cool factor in using the Gyrolab technology, and of course one of the real hallmarks of this technology that I've always just loved was the ability to view those column profiles.
Yeah.
And I can't tell you the number of times I've had discussions with scientists who are dealing with a difficult analytical issue. And I said, well, let's take a look at those column profiles, and you can really see how the ligand binding interaction is happening on the column, and this often gives you a lot of clues about where to go to solve the problem. And I think once people get a hold of that, once they can sort of understand that thinking, it really jumpstarts their ability to do troubleshooting and to build beautiful assays on the Gyrolab platform.
I mean, it gives you so much information on how things are binding in the assay. So, yeah. Which is, as we've said in previous podcasts, is unique to our technology. So,
exactly.
And it's so important, and you can see the affinity of binding, and can really interpret. And also, it's really good for troubleshooting, particularly if you have assay problems as well. So,
yeah, and another application that we've been hearing more and more about is this idea of needing to measure free target assays or free drug assays.
Yeah, and then I mean this appeared in all the user meetings, and it's we have customers moving free assays to China and also free assays in Japan, and it's something that can really only be done accurately currently on the Gyrolab because of the kinetic nature, the fact that you have very, very limited incubation times as part of the assay. It flows over the column. So there's very little chance for dissociation of complex as part of the assay format. And also we've done a lot of work internally and also with our customers trying to not dilute samples as well, because also if you dilute samples, if you apply an MRD, you can dissociate the complex. So combination of incubation time and sample dilution, we've been using free assays in 100% matrix, and we're recommending the high capacity CDs to sort out the matrix problem, because these beads seem to be very good for 100% matrix.
Yeah, when you're doing a free assay in 100% matrix, we've discovered that using the HC bead seems to improve performance for sure. And that's the other piece, I think, that in order to scale that assay, normally you would do an MRD to scale your assay, get it on the curve. You certainly can't do that in a plate without diluting the samples. And so that's the beauty of the Gyrolab system, is that we offer on the HC beads now, the 20, the 1,000, and the 4,000, that really helps to cover the broadest possible range of analyte.
So you can change the analytical range without changing the assay. You just change the CD effectively.
Exactly.
So if you need really low sensitivity, you would go maybe the 1,000 or 4,000. If you don't need a sensitive assay, you may develop on the 20 HC. So
exactly.
Yeah. And I think another area that I have a lot of experience with is immunogenicity, and it's good to see that there are many customers developing immunogenicity assays on the platform, both drug specific, so we're using the drug to actually develop the assay, and also generic assay, which we've released kits, and we've been working with a number of customers. We're kind of the first people to actually release a generic ADA kit, which is good.
We had a number of customers build their own,
but
to have a kit that will allow you to, in the preclinical space, of course, identify when there might be an immunogenicity response that is impacting the PK profile that you're looking at.
And the big advantage in a generic assay is that you're not fighting against the drug for drug tolerance, because you use the drug ADA complex as part of the assay format. So the generic assay is extremely tolerant to the drug because of that. And most specific assays, particularly in the PK profile, it's almost impossible to measure an ADA response. So you can detect ADA a lot easier, and quicker, and with earlier in the PK profile.
Yeah, and so I think we're looking for feedback there, and we certainly have had discussion with folks who are enabling their late stage preclinical work to transition to the clinic by developing a drug specific, traditional ADA assay, but certainly in the earlier preclinical space, or when you're really just trying to understand the circulating complex of immune complexes that might be present in your samples that are impacting PK or other endpoints, then it's useful to have this as an alternative. Again, it's not going to replace the tried and true drug-specific ADA assay, but it certainly has a place, I think, in the workflow.
Yeah, and I think a lot of people do apply clinical approaches to preclinical. Immunogenicity in animals is expected, so there is a less emphasis on necessarily analysing immunogenicity, particularly in the regulated part, unless there's a particular need to do so with anomalous results or change in the PK. So what the generic assay gives is the ability to have an off-the-shelf assay that can be available if you need it, rather than investing lots of time developing an assay which ultimately you may hardly use. So but this is why the generic assay, I think, has an important place, and I can see it being used in regulated studies. So it's not just for discovery type immunogenicity assessments.
Well, I definitely think that's a discussion that we'll be having, because certainly the white papers are not caught up to the developments in this space.
Yeah. I mean, and so lastly, we had a really great discussion here in Philadelphia about affinity assessments using live cells, where really SPR is not an option because you can't incubate the cells on the biosensor. So this is a very exciting application.
Yeah, exactly. And I think the takeaway from that talk was it was an option to do not only in live cells, but a relatively high throughput method. If you're in early discovery, looking at target engagement on the cell for specific antibody constructs or antibody drug constructs, then you can really do a direct assessment of the affinity for your candidate compounds using this approach. So another in the list of hot topics that we saw at the meetings was antibody drug conjugates. Again, my simplified way of talking about this with my family is to say this is like smart bombs for cancer, right? So you have a way to target the drug, the toxin, the payload to the cells of choice. And so it's just a really innovative and cool idea, and it's great that we're seeing that the Gyrolab is enabling a lot of the PK work related to this assays that you need for antibody drug conjugate measurements.
Yeah, and ADC presentations appeared at all the user meetings globally. So I mean, it's very popular in Asia, particularly Japan and China, as well as Europe and in the US.
Yeah,
and there are multiple assays. So you have to measure total antibody, you have to measure the ADC, and also they're normally the free small molecule, but then there was like linker small molecule measurements as well, so the metabolites. So I think it's a very complex area, and there was a whole discussion on the impact of DAR and the amount of small molecules that are attached, and whether the assay has to be able to measure, the ADC assay has to have no bias when you're measuring a DAR 4 or a DAR 8. So there's a huge discussion, and I think Gyrolab as a great platform for ADC measurement. And as was highlighted, and for some customers, their first choice for when measuring ADCs.
Absolutely, and I think that DAR drug antibody ratio is interesting because you have to take into account binding sites available for those, and so that's where you can run into bias issues. And so we often use the affinity purification feature of the Gyrolab to make sure that you're purifying based off of just one of the payload molecules, and then you can come back with your detect. And we heard a really robust discussion in Philadelphia about some of the challenges associated with that, making sure that you have the right reagents to address those potential bias problems that can crop up.
Yeah, with the affinity column, you have a much bigger scope to change the capture concentration than you do with plate-based assays. So, and then this was part of the discussion, and obviously a way of avoiding DAR issues is if you have a single binding site for capture rather than something that can bind differently, depending on how you coat the reagent. So it's a really interesting discussion. And probably a really big, obvious discussion from my perspective was oligonucleotide assays, and we've done the work internally following a master's project, and we've been working with a number of customers, and it's very much a hot topic at the moment. And the use of the Gyrolab to fully automate the hybridization assay is very exciting. So, and this is an area I think we will continue to expand on the Gyrolab.
Yep, and it's just very exciting, and thanks to you, John, for all your hard work and working with the team in Sweden to sort of really push this project along. It's provided a lot of support, and we had a number of customers at the Philadelphia meeting who said they were going right back to the lab to do some of the work and taking off from where you left them with the presentation that you gave. So thanks so much for that.
Yeah, and I think we're achieving really great sensitivity, but also low cross-reactivity, particularly from metabolites. And I think there may be the flow-through technology and the ability to automate the hybridization is kind of unique to our system. So we're looking forward to doing further work in this area, and we are continuing to support customers developing oligonucleotide assays. And another big area is microsampling, blood collection devices, whole blood collection devices. And there's been great advances in this. And I mean, we've been working with Captana and Neoptrix devices and Neoterix devices, sorry. And they're both great devices, and they're working really well. But what we really need to see is a thorough adoption in drug development. And I think they have particular uses, particularly with pediatric sampling, and also rare small trying to collect samples from remote areas, etc.
Right.
And allowing people to collect time points that may be appropriate without a hospital visit. But it's one of these areas that's been talked about for a long time. And it would be great for it to be fully adopted in the drug development.
I agree, and I remember back to the 2008, 2009 Gyrolab Seminar. We were talking about dried blood spot back then. And there are some issues, some technical issues that need to be overcome. But I think the Gyrolab is a perfect pairing for those microsampling devices. And we've had a number of customers, and we have some pretty powerful case studies that show the value of doing microsampling in single mouse PK as an example. And so the face validity of this kind of work has been there for a long time, and just remains to sort of need that extra push to get people to adopt and change some very well-established practices and animal husbandry practices too, to do the actual assays or do the actual collections as part of the drug study in the preclinical space.
I mean, the device, as I said, the devices are great, so you can see them really helping, to have studies involving microsampling. It's just that we just need to know that you don't need to collect samples, both serum plasma and the dried blood spots, or the microsampling devices. So I think they're really working really well.
Yeah. And then, we've seen that growing application to cell and gene therapy space. I think the addition of the kits, the titer and the endonuclease kits, as well as the HEC-293 host cell protein assays that provide folks an off-the-shelf solutions that don't have to do assay development in a space where the time is of the essence and sample volumes, are very precious in terms of production of the viral vectors and so forth.
I mean, again, this is a hot topic from all regions. I mean, there's so many companies working with AAV, for example, and lentivirus, but AAV seems to be the most common, and it's been great. I mean, sample volume is really a restriction with cell and gene therapy, so particularly in the manufacturing of the vector. So our platform is really suited for cell and gene therapy area.
Mm-hmm.
And it's really very much a hot growth area currently.
Right.
I mean, and then finally we had a great talk in Europe. A few years ago, we worked with an academic institution to look at anti-interferon antibodies and their potential part of the pathology of COVID-19, and it was exciting data. And then we now have some work being done in the UK looking at continuing this work, looking at other diagnostic application in HIV and also hepatitis, but also in other inflammatory conditions. So these studying of anti-interferon antibodies could be quite a useful diagnostic, and the Gyrolab is being mainly used for this work.
Yeah,
so.
and I think it's been applied to other diagnostic spaces in exactly the same way, where you're essentially surveying patient samples for the presence of autoantibodies or antibodies to a specific target that are implicated in the disease process. So all really wonderful, exciting work, and we're just thrilled to have the opportunity to interact with all of you who've come to these meetings and shared the work that you're doing, and in that way, John and I get to participate in a broad array of scientific discussions that are, extremely beneficial, and it's just a great opportunity. We really appreciate all of the work that our customers bring to the meeting. Well, here we are, John, at the end of another podcast.
Yeah, I think this is an interesting discussion. I think, as mentioned, we've had some great user meetings this year, so it's good to kind of share everybody some of the discussions that we've had at the meetings.
Yeah, and we've really had a lot of fun doing the podcast. And as you can see, there's a QR code over there. You can. If you're seeing this, obviously you've gotten a link to where the podcasts are, but if you want to get to back podcasts or the archive of all the podcasts that we've done, you can use that QR code or share that with somebody so they can go right to our YouTube channel to listen to any podcasts that you might have missed.
Yeah. Yeah, and I think we are looking for suggestions for future podcasts, and we're likely to have some collaboration with customers who, as I said, we had some great presentations, so we may use the opportunity to use the podcast to actually highlight some of these exciting scientific areas.
I think it's a great idea, and we're going to look at how we can enable that with a little bit of technology, perhaps, and then be looking forward to having some guest lectures or guest folks onto the podcast. I think it'll be a lot of fun, and John and I are having a lot of fun doing it too. So with that, I'm going to say that's goodbye from me,
and it's goodbye from him.
Until next time.